All right, going to give people a few more moments to roll in before we start. Oh, let's get chat up. Hello, testing, testing. All right, okay, looks like we have most of the people that we had for the lecture. So um, I want to get going. Actually, before we get going on today's lab, quick, sorry, um, quick announcement about last week's lab. I screwed up the due date. Unfortunately, I'm sorry about that. It was supposed to be tonight. For some reason, I put in last night. um, if any of you were pressed for, I've changed it to be for tonight. But I also noticed that about half of the class had already submitted their assignments for last week's lab by the due date. Actually, they had submitted. I didn't actually double check the times when they were submitted. So I have graded all of those. If any of you feel you are unable to complete it in time, like you're pressed for time and you didn't do as good a job as you should have, let me know which bits you felt were rushed, and we'll figure out a way of letting you do those bits as you should have if you had had the time to do so. Um, if you had submitted them today and it said that it was late, don't worry about that. They've all been graded as if, the due date was this evening. Um, so yeah, anyway, I'm sorry about that. Unfortunately, these things slip in. Uh, I can make some excuses, and I got some good ones, but still, it's sloppy work. So I'm sorry about that. Does anybody have any questions about the chi-square stuff or the corn genetics from last week's lab before we get moving on this week's lab? Now that could be going over again calculation of expected values, degrees of freedom, how to interpret those charts, things like that. Chance if they have any questions, and please do ask questions if you haven't, or you can also email me them as well if you don't want to say them out in class. What was I going to? Someone else as well. Oh yeah, one of the things that was worth mentioning. So some of you did. I mean, a lot of you did a good job, and I'm glad to see you getting good practice at the chi-square test, because that's a really handy test to be able to use, and you'll be using it later as well. Cool. Okay. Thanks, Angela. One thing that was a little bit surprising though is that some of you found some really crazy non-Mendelian inheritance going on, which shouldn't be the case. Now, I haven't. I graded you based on what you put in the fields for those questions. So if you put, a chi-squared of 20, and degree of freedom 1, then that's highly significant in, and so on. So if you put in that and, followed it through to its logical conclusion, you were graded correctly, right? Because I wasn't so much interested in what results you got, but the fact that you analyzed the results correctly, which is the case for almost everybody. But yeah, it is supposed to be Mendelian. So just keep that in mind when you look at these things, like look carefully at them. And if it's got a little bit of color, just a little bit, it's purple. Otherwise, you get some really weird and wacky results. I think that was really the only thing that kind of came up of note for the calculations. Pretty much follow what's in the lab handout and what we talked about in lab, and you should be fine. But if you do have any questions, email me and let me know. I'm going to be working on stuff pretty much all day today, so I'll be around. Cool. Anybody have any questions about last week's lab before we move on? Okay, do try and keep to time as best as possible, because I know you've got other stuff that you'd probably prefer to be doing. So essentially what we're going to be doing today is kind of a little bit more analysis, really. And so the context for this, let me just bring up where we are. Here we go. The context for this is electrophoresis, right? And electrophoresis is a very handy deal. Hey, come on, give me that back. Come on. WebEx is such a pain sometimes. I'm trying to get the chat box back. There we go. And its main role is to separate molecules, DNA or protein or RNA, by size. And this is really about DNA electrophoresis or gel electrophoresis. And so the deal is here is that if you were in a lab, what you would be doing is running a series of unknown samples, right? So these are going to be bands of unknown size. And essentially, gel electrophoresis separates out that DNA based on size. So the smaller fragments run through faster. The larger fragments take longer to kind of weave their way through the gel matrix. And so when you apply a voltage difference across them, it causes the positively charged DNA to move towards the negative cathode, and they separate out by size. And so you can quite easily visualize like what fragments you have, but there's no way of saying for sure what size they are. And so to get around that issue, we use, and they have a lot of different names. They're called sizing standards here. I call them molecular weight markers because that's what they are. And essentially what you have is another tube or another sample of bits of DNA, right? But these are known sizes. And so you put that in another well and you run it alongside your unknowns, and they'll spread out and you'll get what's called a ladder. That's another word for it, a molecular weight ladder, right? And because all of those sizes are known, because the company that made them presumably knows what they're doing, you can now say, well, this band kind of lines up between these two, and these two are 2000 and 1500, so it's between 2000 and 1500. Right, so that's typically good enough. Like if you're doing a PCR or some cloning or something like that, and you run your sample of interest, and you're looking for something like, say, 450. As long as it's between 400 and 500 on the sizing standard, you call it good, right? But sometimes you want to be more accurate than that. And so the issue here is that, because those standards have discrete steps, your unknown may be in between those steps. And what you need to do is be able to interpolate between those two uh, different sizes and trying to figure out where your thing of interest fits. And so the only way to do that is to calculate a standard curve, right? And the standard curve, if I was a whiz, actually, where's Alyssa? She's really good at Excel. If I was a whiz with Excel, I'll be able to just go tappy, tappy, tap, and I would generate one. But I'm a grumpy old sod, as they say. So I'm just going to draw it instead. Why use technology when you can use stones and chisels, right? So essentially what you have, and this is the basis for all of this lab. So mathematically it's fairly straightforward, but there's often a few hiccups to kind of get around first. Oh, that's a terrible line. No, unfortunately I left the caps off my markers. So they're a little bit dry. I hope they don't dry out because otherwise, oh yeah. Can I enlarge myself? I think you mean embiggen yourself. Active speaker view. Uh-oh. What's active speaker view mean? Ooh, I found an annotate button. I wonder what that does.
It's one of the options for how everyone's like pictures are lined up versus what's being shared. The active speaker view will make whoever is making noise the largest thing on your screen, and then everything else is a little bit smaller.
Okay. Can you repeat that in single syllable words? Because I knows understands. Hang on, there's
There
something. No.
should be like little circles that have different, like there's grid view, there's active speaker video view. If you hover over them, it'll
Ooh,
tell you what the different kinds of views are.
yeah. How's that? Does that work?
Oh, you have to do it like individually. Like if I wanted to see that, like I clicked on it and now you're the main
Oh,
thing that I'm
right.
looking at, and then the screen that you're sharing is like the small tile that is not the main focus.
Gotcha. So each person in here would have to do that themselves. Is that right? Or would I have to do that for you?
Yeah, each person would have to do it.
Oh, okay, cool. Because
That
I
way
just
you
don't
don't
know.
have to stop sharing and then reshare. It's
Oh,
something that we're in charge of.
very handy. Hang on, Angela's added something. Send the line of pics by students by the chat box. Mm. Well, I've done it for you, so you're now speaking, so you're like big and proper, but obviously, I've no idea what you looks like because your video's off. So is that working for you now that the picture is bigger in active speaker view?
It is for me.
Okay.
The
All right.
document's small and I can move it around, so I can move it to another thing on
Now,
my screen.
give me a give me a second. I'll show you. Uh, hang on a second. Unfortunately, I can't share my screen with it in. Okay, so everybody, if you look, see what you're doing. Alyssa was last to speak. So if you have, you should have this on your screen somewhere, hopefully. If you go up to this thing on the right, and this was what I had before, so I kind of, you see everybody down here. Ooh, what's that do? Oh, that's kind of neat. Anyway, go up here and select the one on the left, which says show active speaker video view. And that may fix it for you. Let me know how that works. Is that any better, Angela? Yeah. All right. Well, I don't mind stopping sharing. It's not that big of a deal. Just have to remind me to, yeah, it's recorded. Remind me to check in. Angela, get a bigger laptop. Huge. You should put it on your like a 65-inch TV or something. That'd be hilarious. Anyway, so essentially what you're going to have is what is it going to be? Size. Oh no, pens are running out. Let's see if I can use this one. Oh, that's better. Actually, I think it's the other way around. Distance by, oh no, it is that way around. I'm sorry. Doesn't actually really matter that much. Size in base pairs by distance in centimeters. Right, so essentially those are going to be your two axes. And doesn't help that my laptop camera is like way at the bottom. So on your gel, right, so if that's what you're, I don't know if that's bright enough there. There you go. There's your gel. You're going to have a whole series of bands like so. I'll let you move through. Oh, okay. Well, you can configure, keep on figuring that out. I'm going to kind of channel along with this. Essentially, what you want to do is you want to plot that distance there for this size band, right? So that's a big band, and it's going to move a small distance, right? So you're going to plot that like so. See if I can stretch my legs out and put this even further out. There you go. Now you're going to do the same for this one. All right, that is that distance. It's always from the beginning. This is where you load the DNA. And essentially what you're doing is you're plotting out how far each band moves, which is this distance, by the size of that band, which you know because I give you that information in the handout. So for the next one, that's going to be a little bit smaller, and it's going to move. And then you do that for all of these. And what you'll find is that it's not a perfect straight line, right? And so this is essentially, you're making a standard curve, right? Because you're going to be plotting your known values, right, which are the sizes, against the discovered values, which are distance. And if I can do you got to do this with a ruler. I'm not, because I don't have one to hand. Essentially what you do is you fit a line to that. This is essentially, remember at the beginning when I said you need to interpolate between the two different values, right? Say for this band here, right, that one, it lies between these two. So it's very hard to give an exact number, say, oh, this is, I don't know, 9,000, right? If this is 10,000, 8,000, because the distance that they move may not be perfectly linear, right? And so how do you figure out the size of that band, right? That's something in between. The first thing you do is you interpolate between them by drawing a line of best fit. And this is basically a line which I've tried to do a decent job of it here. It fits, it's as close as possible to all of the points. Now it can't be a wiggly line because that's not going to work. It has to be a straight line. And so you can see that if I had instead done that, I don't know if you can see this super well, it's kind of a little dim in here, perhaps because of the light behind me. Let's get this shut. Yeah, maybe that helps a little bit. So now I've tried fitting this line to just these 2 points, right? Because I want to find out the size of that one. But as you can see, it's really far from these two, right? So it's not a well-fitted line. So you need to, and likewise, you could do the same over here, right? For example, right? That's not your standard curve. Your standard curve is the one that goes through as close as possible to every single line, every single point, sorry, that would be described by that line. Okay, give you a few moments to cogitate on that. I hope my black pen comes back. Be sad if it doesn't. Okay, anybody got any questions or queries, or what language are you talking? This makes no sense. It's really more math, you know. As annoying as math is, it's kind of hard to avoid. Yeah, yeah, you get out there, Alyssa. You math lover, you. Actually, I shouldn't speak too much. I love playing around in Excel, even though it's technically not math. Anyway, so you want to draw that line, and you're going to have, yeah, let's get that out of the way. You're going to have two of these ladders, right? You're going to have one by Promega. That's the one that has a whole ton of bands in it. And you're going to have another one by a company called Edvotech. Both of those pictures are in the handout. Those are your two known sets of bands. Again, you're going to be plotting the distance that each of those bands moves, like from over here on the left-hand side, that one, by distance. And you're going to measure that from a fixed point, right? And that point, because you may need to actually draw this, like print this out on a piece of paper, may help. Whatever that point is, you have to do that for all the bands. So I've chosen this point here, which is the well. Could be the top of the piece of paper. Could be just a line you draw somewhere. It really does not matter. As long as you measure from here to here, then from this same point to here, this same point to here, the same point to here, right? You have to measure from the same point to be able to get that standard curve. Right, once you do that, now you can figure out your unknowns, because as you can see, this line here, it bridges all these points. So if you have any unknown which lies between these points, you can calculate its size, right, by using that standard curve. So let's see if I'm going to... All right, let's get rid of that green because I don't want to confuse you. Okay, so let's measure this one, right? This is one of our unknowns. Right, so I'm going to measure this. You do it with a ruler. You do it with your fingers if you want. That's kind of crude, but rulers generally better because they, centimeters are the same size wherever you're at. And now you take that value and you plot it here. So where would that be? That would be about there on this axis, right? So we need to find out two bits of information. We need to find out the distance this band travels, and we want to find out the size of that band. So let's stick this on. A little overheating here. We know the distances traveled, and so we're going to use that information and this standard curve to work out its size, the unknown, the second piece of information. And the way you do that is you get it where it is here. This is kind of like using that chi-square chart for figuring out p-values. And you draw it up until it hits that standard curve. And then you see where it intersects the y-axis. And the y-axis is size. And so let's say this is 10,000. And this is 8,000. Like so. Now, and this is, what's that? 9,000. Now you can say, oh, I reckon that is, say, 9,100 base pairs. That's the units. So this particular band over here is 9,100. And then you do the same for this one. like so. And then you do the same again. That would be over here somewhere, right? You draw it up until it hits the standard curve and across until it hits the y-axis. And then you read that value from the axis. And you're going to be doing that for two different, pardon me, markers, the Promega marker and the Edvitec marker. So you need to draw two different graphs. You can either do this in Excel if you're handy in Excel, or you can do it on paper. And let's hang on, got to share content screen. So if you go to the lab, right, you'll see here's the lab handout, right, which you'll be working from, and that will take you through this whole process too, right? So it's essentially the same as what I'm telling you right now. It also has the pictures. I don't have them out here. The pictures of the two markers, and so this is the gel picture that you will be using. This is essentially what you would be producing yourself, but unfortunately, because we're not in the lab, you can't. And this is the Promega ladder, so you can see it's got an absolute ton of bands in it. I think that's 10 and 8,000. This is probably 1,000. That's 5,000 maybe. And you have another marker which is a lot simpler and only has, I think, 7 bands to it. These are your unknowns here, right? And your job will be to draw two standard curves, one for this one for this one. You can use this semi-log paper if that helps. It's up to you. Hang on, let's make sure. Oh, hang on. Sorry, Angela, my chat disappeared when I shared my screen. Yeah, the far right ladder is Edvitec, but if you go, which one is it? This one. Yeah, I didn't do like a bang up job of labeling the lanes because I was a little lazy. But left to right, it's Promega unknown 1, 2, 3, and the Edvitec size and standard. And so this is what the Promega one looks like. Oh, that's actually 3,000, that bright white one, sorry. And this is what the Edvitec one looks like. So it gives you the known sizes. These ones are nice and neat, round numbers. These ones are not. So you might need to round a little bit when you start doing your standard curve. So yeah, so this is the gel picture you're going to be using. It's also in here, right? But I uploaded it separately just in case you want to blow it up and print it out on a piece of paper or something like that. And you can draw anywhere in here a line, straight line, parallel to the top of the image, and measure from that, right? So it's up to you what you do or where you do it, whether you use like pencil and paper or if you use a computer or whatever. But you have to measure all of these bands from the same point. And then I've included here, this is the actual unknown sizes. So what I want you to do, don't just look at these and copy them out because that will defeat the object. You need to do a graph like this for each of these two markers. You need to measure the distances for all three of these unknown lanes, right? So this one has 1 fragment, this one has 2, and this one has 3. And then I want you to figure out the sizes of these unknown fragments, right? And basically compare them to the actual sizes and tell me which ladder you think is better, which standard curve is more accurate, right? So again, measure all of these bands, as many of these as you can, right? You don't need all of them because some of them are fairly close together, right? And some of them are a little faint. So measure as many as you can, plot that out, and then figure out using that standard curve from this marker what these sizes are. Then you do the same for this one. Measure all of the distances of these bands, make your standard curve, figure out the unknown sizes from that. And then, actually, I can probably even show you this because there aren't any yes, no answers in here. And I have checked the due date for this one. It is correct, so I didn't make that mistake again. So essentially, I'm simply asking you to do what you did. Hey, I've tried this twice already. What you're being told to do in the handout, right? So you need to do a plot and your standard curve, right? And take a picture or save the file and then upload it to this, right? So that's for your ProMega size and standard. Repeat the same for your Edvatec size and standard. Load it up in here. And I go through these manually and I look at them all and, score it out of 15. As long as your axes are labeled, your line is straight, you're kind of good, essentially. And then your third answer is simply, there we go back. The distances of these, so it's kind of just forcing you to write down the results that you're getting, right? So the distance for unknown 1, unknown 2, A and B, unknown 3, A, B, and C, essentially, right? That you need to put in to this answer here. Then you're going to calculate sizes of those from your ProMega standard curve. That goes in here. Your Edvatec. Oh, that didn't come out very well. Might need to just, oh, it's in there anyway. From your Edvatec and add them there, right? Again, for unknown 1, 2, and 3. And then finally, just a little bit of which do you think is better and why, right? Doesn't have to be a very long essay or anything like that, just a few sentences. If you did find a difference, which was the better, which was closer to the actual values, and why you think that was so. And that's pretty much it, right? And so here are the actual values, right? So that's what you're going to be comparing your calculated values to. Any questions? Because that's pretty much it. It will take you a little while, just because it's a little time consuming to draw those graphs. And the only thing that sometimes students get a little hung up on is this part. The whole get your unknown 1. Let's say this is unknown 2, right? Because unknown 2 has 2 fragments. Unknown 2 fragment 1, you put the size down here on your distance traveled axis. Draw a straight line, pardon me, up until you hit the standard curve or your straight line, and then draw a straight line across. That's your size. And then you repeat that for the other fragment in unknown 2, you get your second size down here. Right, so for unknown 3, you got 3 different fragments, you'd be doing 3 of these. And then compare these values that you get from your gel and from your 2 different graphs, and compare those to the values you see here. And that's it. So, no, it depends how quickly you are at graphing, but it'll take you an hour to 2 hours. Hopefully not more than that. If you do have any questions, though, either send me an email or we can go over them in the beginning of lab on Thursday, as the deadline is Thursday evening. I think that's pretty much it. How do we find the distances of the unknown? So that you do the same as you do for each of, here we go. Each of these. So you're going to be finding the distances of each of these bands in your marker, literally with a ruler. I wish I brought one up with me. Right, you're going to measure along, that's that far. That's that much further. Those are the values that you're going to be using to plot this. The distances of the unknowns are worked out exactly the same. You simply measure them with a ruler from your top point where you're measuring from. And again, it doesn't matter where on the picture you draw that line. Top of the piece of paper, maybe, would be simplest. And then you measure from that point to the first unknown band. And that's the value that you then put in down, where am I at? Down here on your, what is it, x-axis. Does that make sense? Sorry, Gabrielle, I'll just be getting to you in a second. Just seeing if I answered that student's question properly. Awesome, cool. But again, once you try, if it still doesn't make sense, just send me an email and I'll address any concerns you have. So yeah, plotting the EdvoTech is exactly the same, Gabrielle. So you're going to have, where are we at? Two different markers doing two graphs. You're going to plot it once. You're going to calculate all the distances, right? Your marker distances for these bands. Hopefully you can see what I'm pointing at. That's going to be your ProMega standard curve. And then on the second graph, or piece of paper or whatever, you do the same for the EdvoTech marker on the right. They're basically just different molecular weight markers. They're going to behave the same. So they're going to travel the same size will travel the same distance, right? That's pretty linear. That's how you're getting this standard curve. It's just the sizes are different, so the distances are different. Yeah, that's correct, Angela. You'll be making two different graphs. Technically, you can do it on one graph, but it's way easy to get confused because you're going to have two different standard curves on the same graph. So I'd recommend just doing two different graphs. And then once you've measured the distances of the unknowns, right, these ones, you're going to use the same values on both graphs. So, if this is your ProMega graph, you're going to be putting in the unknown distances down here and reading off the sizes here. Let's say I just rub all this off and there you go. Pretend that it's a different graph. Then you would use the same unknown fragment distances that you used before, put them in down here, and then you read off this line, which presumably would be a little bit different, this standard curve. Yeah, so you do two graphs, one for the ProMega, one for the EdvoTech. You measure the unknown travel distances, and again, you're going to have to put that into the assignment on Blackboard. And then you use those unknown distances and the standard curve to figure out the sizes of the unknown fragments. Yeah, so this red line is the standard curve. So these points here, right? So these kind of like stars, those are the individual points of distance and size for each marker. So for example, let's go, crab sticks, where are we at? Let's use an actual example. And I'm just going to go through, even though you got it, I'm just going to kind of continue on a little bit just to make sure everybody else has. So for example, on the EdvoTech marker, it is some like funky ass number. it's 6,751 bases. Why it's that number exactly, I don't know, but that's the topmost one, right? And the bigger ones travel less distance. Now, when you see on here on the gel picture, you're going to measure, say, from the top of that picture to that band there, either the top or bottom, it doesn't matter. Let's say that is, what does that look like? 3.5 centimeters on my laptop. So let's get rid of this. Let's say we're doing it for the Evertec. And again, kind of laboring the point a little bit, but I really do want to make sure that you all get it. So, I'm so pissed off I left my cap off my black marker. So let's say we've got 10,000, and again, this is DNA size, 8,000, 6,000, 7,000. I don't know, something like that. It's actually a log scale, which makes it a lot easier to use. So the Evertec one is 6,751. So that's going to be about here. And let's say this is 3, 4, 5, 6 centimeters down here. So that would be the first data point from the Evertec one. And then you just repeat this, right? You go back to here, the next one is 3,652. That's say 4 centimeters, right? So that's going to be, again, it's a log scale, makes life a lot easier. And then you draw a line through that. And so that you have to do for both of the markers to generate those two standard curves. Oh, was she complaining? Got another cat. She was meowing to come in. Okay. Did that help everybody? Hopefully that makes sense. Cool. But again, if you do have any issues, questions or problems or whatever, just send me an email. And I'm also more than happy to arrange just like one-on-one WebEx meetings with anyone to go over this stuff in person. Sometimes it's easier to do that in person than it is by email, to be honest, right? Because you get to kind of weasel out whatever sticky points are causing problems. So if you do want to do that, just let me know and I'll arrange that as well. My days are pretty easygoing, generally speaking. Cool. Okay, and that's it for lab today. So hopefully you'll be able to use the rest of the time allotted. Oh, cool. Thank you. I'll have a look at that in a second. Actually, hang on. Don't leave the meeting. I'll have a look at it and then we can talk before you go. Yeah, so any questions or queries or whatever, just send me an email and I'll get onto those straight away. All right, cool. Thanks very much, everybody. So I'm going to end, stop sharing. And stop recording.